Whether you are a medical student prepping for rounds, an oncology professional deep in the literature, or, just a dedicated learner of the biological sciences, you're already pretty intimately familiar with the core dilemma of systemic cancer therapy.
Right, the whole carpet bombing approach.
Exactly. I mean, we've relied on systemic carpet bombing for decades. You flood the vascular system with highly toxic cytotoxic agents, and you're really just banking on the therapeutic index.
You're just hoping to induce apoptosis in those rapidly dividing malignant cells, just a little bit faster than you destroy the patient's healthy epithelium or your bone marrow.
Yeah, it's a war of attrition, literally.
Oh.
But the paradigm is shifting. We're moving toward, well, a microscopic sniper approach.
Which is fascinating. The goal now is to deploy a delivery vehicle so small it just navigates the systemic circulation completely undetected.
Right, and it localizes exclusively to the tumor microenvironment, releasing a lethal payload directly into the cytosol of the malignant cell.
But getting that microscopic sniper from the injection site to the intracellular target, well, it requires bypassing some of the most aggressive and highly evolved biological barriers in human physiology.
It's insane what these particles have to survive, which is why today we're taking a deep dive into the mechanics of that exact challenge.
And we're pulling this from Lautfei H. Madkor's really comprehensive research, his textbook on nanoparticle-based drug delivery in cancer treatment.
Yep, and for this deep dive, we are going to bypass the basic biology and just jump straight into the engineering of nanomedicine.
So we're talking about how we construct these carriers, the physics of their stealth capabilities, the specific inorganic and organic materials utilized.
And how we're currently engineering payloads to circumvent multi-drug resistance mechanisms, like efflux pump-up regulation.
Right, because, the primary hurdle is simply keeping the drug in systemic circulation long enough for it to even reach the target.
Because a bare chemotherapeutic molecule is just completely at the mercy of renal clearance and hepatic metabolism.
Exactly. So by encapsulating that drug within a nanoparticle, we can dictate its pharmacokinetic profile. But, and this is a big but, only if we strictly adhere to a very narrow physiological size constraint.
The dimensional sweet spot is incredibly tight, isn't it?
Oh, absolutely. We're dealing with a therapeutic window that's generally between 10 and 100 nanometers.
Because if we engineer particles smaller than 10 nanometers,
It is susceptible to rapid renal clearance.
right, the glomerular basement membrane in the kidneys operates like this microscopic sieve.
Yeah, the fenestrations in the endothelial cells and those podocyte slit diaphragms, they will readily filter out pretty much anything below that 10 nanometer threshold.
So the nanocarrier is just cleared right into the urine within minutes.
Yeah.
Which makes it therapeutically useless.
Completely useless. But on the other end of the spectrum, if you engineer a complex carrier that exceeds, say, 100 to 200 nanometers, you trigger rapid clearance by the MPS.
The mononuclear phagocyte system.
Right. So the Kupffer cells residing in the liver sinusoids and the macrophages in the spleen's marginal zone. They are exceptionally efficient at sequestering circulating particulates of that size.
Because they just recognize them as foreign bodies, right?
Exactly. They recognize them, engulf them via phagocytosis, and just remove them from circulation.
Wow. Okay, but even if we nail that, like 50 nanometer sweet spot, size alone doesn't actually prevent MPS clearance, does it?
No, it doesn't. The surface chemistry of the nanoparticle is just as critical, mainly because of opsonization.
Right, opsonization.
Yeah, the moment a hydrophobic nanocarrier enters the bloodstream, circulating serum proteins, opsonins, adsorb onto its surface.
They essentially coat the particle. It's like tagging it with a biochemical neon sign.
That's a great way to put it, a neon sign that just screams at the macrophages to initiate phagocytosis. So to counter this, we rely heavily on pegylation.
Pegylation, which is essentially the gold standard for nanoparticle stealth, right?
Pretty much. We covalently graft polyethylene glycol PEG chains onto the surface of the nanocarrier.
And the mechanism there is steric stabilization.
Right. PEG is a highly hydrophilic polymer. Its repeating ethylene ether units form extensive hydrogen bonds with all the surrounding water molecules.
So it creates this dense, thermodynamically stable hydration shell around the particle.
Exactly. So when those circulating opsonins attempt to approach the nanoparticle surface, they physically compress that hydrated PEG layer.
Which creates a massive entropic penalty, right? Because the compression restricts the conformational freedom of the polymer chains.
Yes. The proteins are literally repelled by the thermodynamic resistance of the water molecules. It renders the nanoparticle practically invisible to the immune system.
So it's kind of like a sci-fi spaceship navigating a hostile galaxy. It has to be big enough to avoid falling through the asteroid gaps of the kidneys.
But small enough and cloaked via pegylation to avoid the radar of the liver's macrophages.
That's amazing. But I know there's also a biofabrication angle here too, right?
Yeah, actually. While synthetic chemistry gives us pegylation, there's a parallel movement utilizing biofabrication. We can leverage the natural metabolic pathways of microorganisms to synthesize our metallic nanoparticles.
Wait, really? Like bacteria building our nanomedicine?
Yeah, certain strains of cyanobacteria or even fungi like Fusarium keratoplasticum, they possess extracellular enzymes that naturally reduce metal ions.
Oh, wow. So you just feed them specific precursors?
Pretty much. You harness their biological machinery to churn out highly uniform silver and gold nanoparticles, and they're stabilized by natural fungal proteins rather than synthetic polymers.
That is wild. Okay, so assuming we have this perfectly sized cloaked nanocarrier successfully circulating in the plasma, surviving the journey is only half the battle.
Right, because systemic circulation is a vast network. The carrier still has to preferentially accumulate in the tumor tissue itself.
Which brings us to the localization problem. And I assume this is where the EPR effect comes in, the enhanced permeability and retention effect.
Exactly. You see, the architecture of tumor vasculature is notoriously chaotic. Driven by the tumor's hypoxic environment and the overexpression of VEGF, tumor angiogenesis occurs at a frantic pace.
They're building blood vessels way too fast.
Far too fast. So the resulting capillary beds are highly tortuous, disorganized, and structurally defective. The endothelial cells lack a continuous, smooth muscle layer.
And they have those massive gap junctions, right?
Yes, fenestrations ranging anywhere from 100 to 600 nanometers wide.
So because our engineered nanoparticles are around 50 nanometers, they easily extravasate through those huge gaps right into the tumor interstitium.
But the retention half of the EPR effect is just as vital. Solid tumors typically lack functional lymphatic drainage.
So the high interstitial fluid pressure coupled with the missing lymphatic vessels just creates a sink.
Exactly.
Yeah.
The nanoparticles are pushed through the leaky vasculature and they just become trapped in the tissue.
Okay, but I want to introduce some friction here for the listener. Because the EPR effect is the workhorse of passive targeting.
It is, yeah.
But relying solely on passive accumulation has massive clinical limitations. If passive targeting successfully localizes the drug to the tumor bed, why do we dedicate immense resources to active targeting?
Well, the EPR effect varies wildly depending on the tumor type, its anatomical location, patient biology.
Right, but if the concentration of the cytotoxic agent is high enough in that surrounding interstitium, shouldn't diffusion gradient kinetics just drive it across the cell membrane?
Diffusion works for certain lipophilic free drugs, but remember, our payload is encapsulated in a highly stable hydrophilic nanoparticle designed explicitly not to break down easily in plasma.
Oh, right, so getting it to the neighborhood isn't enough.
Exactly. The EPR effect gets the drug to the neighborhood, but active targeting is required to get the drug inside the house. We need the entire carrier to be internalized by the malignant cell before it releases the payload.
So how do we get it inside?
Active targeting overcomes the cell membrane barrier. We functionalize the surface of the nanocarrier with specific targeting ligands.
Things like folate, transferrin, or RGD peptides.
Right. Ligands that have a really high binding affinity for receptors that are heavily overexpressed on the tumor cell surface.
A classic target would be those alpha V beta 3 integrins overexpressed on tumor endothelium, right?
Exactly. So when the RGD peptide on our nanoparticle binds to that integrin receptor, it initiates receptor-mediated endocytosis.
So the cell membrane physically invaginates, enveloping the nanoparticle into a clathrin-coated pit and just pinches off to form an intracellular endosome.
Yeah, we've successfully hijacked the cell's own nutrient scavenging pathways to pull our Trojan horse directly into the cytoplasm.
That's brilliant. Okay, so now that we know how they arrive and enter the cell, let's talk about the Trojan horses themselves. What are these vehicles actually made of?
Well, the nanocarrier arsenal is broadly divided into organic and inorganic platforms. The organic carriers are definitely the most clinically mature.
Like liposomes.
Yes, liposomes are the foundational technology here. They're spherical vesicles composed of phospholipid bilayers. They are perfectly suited to encapsulate both hydrophilic drugs in their aqueous core and hydrophobic drugs within the lipid bilayer itself.
Doxil's the classic textbook success story for liposomes, isn't it?
Oh, absolutely. By encapsulating doxorubicin within a pegylated liposome, the pharmacokinetics shift drastically.
Because usually, free doxorubicin causes severe cardiotoxicity. That's the dose-limiting factor.
Right. But the liposomal formulation restricts the drug to the vascular compartment until it accumulates via the EPR effect. It drastically reduces off-target myocardial damage.
We also see massive clinical utility in albumin-based carriers, right? Like Abraxane.
Yes, Abraxane binds paclitaxel to native albumin. That actually eliminates the need for toxic solvents like Cremifor EL, which previously caused severe hypersensitivity reactions in patients.
Okay, so liposomes are basically like water balloons delivering a payload. What about the inorganic platforms?
With inorganic platforms, the engineering shifts toward theranostics.
So therapy plus diagnostics.
Exactly. Integrating targeted therapy and diagnostic imaging within a single platform. Gold nanoparticles, or GNPs, are highly versatile in this space. They exhibit this phenomenon called surface plasmon resonance.
Right, so when you irradiate these gold nanoparticles with near-infrared light, which penetrates human tissue pretty well.
The conduction electrons on the surface of the gold lattice begin to oscillate collectively, and that oscillation rapidly decays through non-radiative pathways, releasing immense amounts of thermal energy.
So we use that for targeted photothermal ablation. You inject the GMPs, wait for them to accumulate via EPR and active targeting, and then apply a near-infrared laser.
To literally cook the tumor, you denature the cellular proteins and induce localized necrosis. Plus, GNPs are highly radiopic.
Meaning they provide massive contrast enhancement for CT imaging.
Yep. And their high atomic number significantly amplifies the localized dose of therapeutic X-ray radiation, which drastically improves the efficacy of standard radiotherapy.
Okay, gold is like a microscopic multi-tool, doing double duty as a beacon and a weapon. But I have to push back on the inorganic platforms here.
Oh, how so?
We spend so much time worrying about the pharmacokinetics of the payloads. But what about the carriers themselves? Gold nanoparticles, quantum dots, mesoporous silica. These are heavy metals and robust crystalline structures.
That is a very valid point.
If they don't degrade metabolically like a lipid bilayer does, aren't we risking massive long-term toxicity? Like via accumulation in the hepatic or renal tissues?
You hit the nail on the head. The toxicity of inorganic platforms is the primary bottleneck preventing widespread clinical translation right now. Quantum dots, for instance, offer unparalleled fluorescence for intraoperative tumor margin mapping.
But their cores are often made of cadmium or selenium, right?
Yeah.
Highly toxic heavy metals.
Exactly. So to mitigate this, engineers have to apply robust, biocompatible polymer coatings to prevent heavy metal ion leakage.
What about mesoporous silica nanoparticles? MSN.
With MSNs, the toxicity profile is actually much more favorable. Silica eventually dissolves into silicic acid in the body, which is renally cleared.
And MSNs are fascinating from a fluid dynamics perspective, like a microstatic sponge. They have this honeycomb-like porous architecture.
Yes, they're synthesized via sol-gel chemistry. And because of the massive surface area to volume ratio and the capillary action of those microscopic pores, they achieve exceptionally high drug loading capacities.
You can just pack a massive volume of hydrophobic chemotherapeutics into the silica matrix.
But to circumvent carrier toxicity entirely, the field is actually advancing toward carrier-free nanodrugs.
Wait, carrier-free, how does that work?
Through supramolecular chemistry, we can engineer pure prodrug molecules that spontaneously self-assemble into stable nanoparticles via non-covalent interactions.
Like pipey stacking or hydrophobic forces.
Exactly. This yields a nanocarrier comprised almost entirely of the active pharmaceutical ingredient. It pushes drug loading capacities toward 100%, completely eliminating the need for extraneous carrier materials.
That is incredible. Okay, let's transition from the vehicle to the payload itself. Because delivering a standard cytotoxic agent into the cytosol is often insufficient against advanced malignancies, right?
Unfortunately, yes. We run headfirst into multi-drug resistance, or MDR.
Right. A patient might exhibit a phenomenal initial response to chemotherapy, but inevitably a subpopulation of tumor cells adapts, survives, and repopulates the tumor.
With a highly resistant phenotype. And the molecular mechanisms driving MDR are incredibly robust. The most prominent is the upregulation of efflux pumps.
Like the ATP-binding cassette transporter family, P-glycoprotein specifically.
Yes. These transmembrane proteins utilize the energy from ATP hydrolysis to actively pump lipophilic drugs right back out across the cell membrane against their concentration gradient.
So the moment the free chemotherapy molecule diffuses across the lipid bilayer, The P-glycoprotein pump undergoes a conformational change and just ejects the toxin back into the extracellular space.
Before it can even intercalate with the DNA or disrupt the microtubules. It's incredibly frustrating.
And the second major MDR mechanism involves the upregulation of anti-apoptotic pathways, right?
Right. Cancer cells frequently overexpress proteins like BCL2. So even if the cytotoxic drug successfully damages the cellular DNA, BCL2 stabilizes the mitochondrial outer membrane.
Which prevents the release of cytochrome C and completely halts the apoptotic cascade. The cell sustains massive DNA damage, but stubbornly refuses to die.
But this is exactly where the specific intracellular routing of the nanoparticle bypasses the defense mechanisms. Remember, receptor-mediated endocytosis.
The Trojan horse effect.
Exactly. Because the nanocarrier is brought into the cell encapsulated within an endosome, it completely bypasses the transmembrane efflux bumps. The P-glycoproteins are positioned to intercept molecules diffusing through the membrane, not large biological vesicles.
So by the time the nanoparticle undergoes endosomal escape, which is often triggered by the acidic pH of the late endosome degrading the carrier matrix, it releases its payload deep within the cytosol.
Or directly adjacent to the nucleus. We've effectively smuggled the drug past the perimeter guards.
But the most elegant solution to MDR involves co-delivery platforms, doesn't it?
Oh, absolutely. The pinnacle of current research. We are no longer just delivering a cytotoxic agent. We are simultaneously delivering a genetic modulator, specifically small interfering RNA or siRNA.
Capitalizing on the RNA interference pathway.
Right. We designed the siRNA to specifically match the messenger RNA transcript that encodes for the P-glycoprotein efflux pump or the BCL2 anti-apoptotic protein.
So when the nanoparticle releases its payload into the cytosol, the siRNA is incorporated into the RASE complex, which then locates and cleaves the target mRNA.
We are fundamentally cutting the supply lines, silencing the expression of the defense proteins at the translational level.
And once the cell's defensive architecture is genetically turned off, the co-delivered chemotherapeutic agent is released to operate unimpeded.
Exactly. There are real-world applications showing this right now. Clinical trials utilizing DOPC liposomes to systemically deliver siRNA targeting the EphA2 oncogene. It is demonstrating just how potent this 1-2 punch can be in overcoming previously intractable resistance.
But I have to ask, since RNA is notoriously fragile, is this where the physical protection of the nanocarrier is most critical? Because if you were to inject bare, unencapsulated siRNA directly into systemic circulation,
Serum RNases would degrade it within seconds.
Mm.
The phosphate backbone is highly susceptible to enzymatic cleavage, plus its strong negative charge prevents it from crossing the cellular lipid bilayer anyway.
So the nanocarrier provides absolute steric protection from serum nucleuses while simultaneously facilitating intracellular delivery.
It is the ultimate synergy between materials science and molecular biology. The physics of the carrier solves the systemic delivery challenge, and the genetic payload dismantles the cellular resistance mechanisms.
Amazing. As we synthesize the mechanical and clinical concepts we've explored today, I want to leave you, the listener, with a clinical thought exercise to really reinforce these targeted delivery mechanisms.
A little review question.
Exactly. When you are evaluating the pharmacokinetic profile of a novel nanodrug on the ward, consider the distinct physiological barriers bypassed by passive versus active targeting. If an attending asks you to articulate why a purely pegylated liposome might exhibit high tumor accumulation but poor therapeutic efficacy compared to a folate-conjugated liposome, walk through the exact biophysics.
Think about the physical differences.
Right. Consider how the fenestrations of the EPR effect drive interstitial accumulation, while the requirement for receptor-mediated endocytosis is absolute for bypassing the lipid bilayer and delivering the payload into the cytosol. Understanding that the distinction dictates the clinical utility of the drug.
And to look beyond even the current clinical trials, I want to offer a final forward-looking concept from Madcor's text to ponder.
Let's hear it.
We framed this entire discussion around discrete, independent nanoparticles. But the next frontier involves supramolecular networks.
Wait, networks? Like they communicate?
Exactly. Researchers are exploring nanoparticles that use molecular interactions to communicate and link up after they enter the body. Imagine deploying swarms of nanocarriers that circulate independently, localized to the tumor microenvironment, and then dynamically self-assemble into complex macroscopic structures.
That sounds like science fiction.
It does, but it's happening. They assemble in response to localized biochemical triggers from the disease itself, like matrix metalloproteinase activity or localized acidosis.
So we're looking at a near future where the therapeutic vehicles dynamically adapt their physical structure and release kinetics in real time based on the specific pathophysiology of the tumor they have infiltrated.
It is a staggering leap from the systemic toxicity of traditional chemotherapy.
Truly mind-blowing. Well, thank you for joining us on this rigorous deep dive into the nanomedical frontier. Until our next analysis, keep questioning the mechanics.